Hey guys, welcome back to my channel. This is the Cortex, and today we will be continuing with IGCSE CAIE biology, and this chapter is enzymes. This is part 1 of part 2. This is a this is a easy chapter but a big chapter, so let's get on with it. Now this is the learning objective. This is part 1. So in this we will learn chemical reactions in a cell, catalyst and enzymes, enzyme controlled reactions, classification of enzymes and examples of hydrolases, industrial uses of enzymes, characteristics of enzymes, some of them, and lock and key hypothesis. So let's continue with it. We have a lot to do. Chemical reactions in a cell. As we learned that a cell cannot live without any chemical reaction, it has to make products and even eliminate waste products. So let's read. Chemical reactions greatly depend on the pressure and temperature of the atmosphere. Now see, remember we did this in chemistry in kinetic particle theory. If we see the combustion or burning of sugar, it requires a very high temperature. How do cells carry out these reactions in so low temperatures? Because as compared to sugar, how much temperature, very high temperature it needs, with boiling temperature, how are cells able to reach that temperature? They respire in numerous steps, due to which small amounts of energy is released, which is trapped in high energy molecules like ATP, which are used to carry out even more reactions. Now what this says, when it is burned, all the energy, the respiration, you could say, but I would say call it combustion, like in when you do boiling, when that occurs, all of the energy is released together, whereas in a cell they respire in numerous different steps. So in different steps, very small amount, different amounts of energy is released, which is trapped in high energy. Now what happens with that small amount of energy? Obviously we don't want it to go to waste, so it traps it in molecules of ATP, which are adenosine triphosphate, remember in the first chapter of cells, which are used to carry out even more reactions, because they are basically like a battery or like a chemical potential energy. These multi-step reactions are only possible due to enzymes, because enzymes carry out reaction and they need a lot less activation energy. You will learn what is activation energy with me in the coming slides. Catalyst. Now what is a catalyst? You should know what is a catalyst before learning what is an enzyme. A catalyst is a substance that can be added to a reaction to increase the reaction rate without getting consumed in the process, which means it increases the rate of reaction without finishing. Catalysts typically speed up a reaction by reducing the activation energy or changing the reaction mechanism. Now what is activation energy, you might ask? Activation energy is the energy needed to carry out the reaction. It lowers it down, which means, as you can see over here, it even says that, which means it in numerous steps small amounts of energy is released. So that's how it works. But there's a huge difference in catalyst and enzymes. We'll discuss it. When potassium chlorate is heated strongly, oxygen is given off and potassium chloride is made. But if you add a little bit of manganese oxide, so the reaction occurs even more quickly. Thus manganese oxide is a catalyst, because it does not finish and also it makes the reaction occur quickly. It does not alter the products, it just makes it happen even more quickly. You see, this is the basis of the enzymes and the catalyst. See, this is the potassium chlorate molecule attaches over here and then it breaks. Now we will study in detail of this in the enzymes. Now what are enzymes? You see, they are globular proteins coiled into very precise three-dimensional shape with hydrophilic side chains making them soluble. So what we learned from this, that enzymes are soluble first of all, and they have a very precise three-dimensional shape. Now let's see the correct and the best meaning you can write. Enzymes are biological catalysts. You see, now they are not only catalysts, they are biological catalysts, which is the one of the biggest differences in catalysts and enzymes. That are made of protein, sometimes RNA, which is ribonucleic acid, which we will study in further coming chapters, and they alter the rate of reactions without being chemically changed at the end of the reaction, which means they are not altered and they can be used again. They catalyze many biochemical processes in a living cell over a very narrow temperature range because the energy is released in short amounts in many different intervals. Enzymes speed up reactions by lowering the activation energy of the chemical reaction. Now see, in any chemical reaction, molecules must reach a certain energy state before reaction is possible. This is known as the activation energy, it says over here. Now what certainly happens? This causes a redistribution of electrical charge which strains the chemical bonds within a substrate. Substrate is the molecule that an enzyme acts on. Therefore, more molecules can cross the energy barrier and the rate of reaction is faster. Now the electrical charge, because you know chemical bonds very related with electrical charges, we will learn this in chemistry, but what happens, it redistributes the electrical charge. That's why it makes the reaction happen faster, because more molecule can cross the energy barrier. This might sound a little confusing, but it is not a really important point, but you should know. Enzymes are also used to break down carbohydrates, fats and proteins in labs, as it requires complex apparatus without them. Now labs require very complex apparatus to carry out the breakdown of carbohydrates, fats and proteins, but an easy way out of this is to use enzymes. All enzymes have unique shapes. Enzymes can either break down complex substances into simpler substances, which means breaking them down, which is known as catabolism, or building up complex substances from simpler substances, known as anabolism, which either means that they can break a substance or create a substance. That depends on them though. They can only increase rate of reaction and not amount of product. Okay, you see, this is an enzyme very similar to the catalyst. This is, for example, a fructose molecule. It is then attached to the enzyme and then it breaks. We will learn in detail how it attaches and what is the hypothesis related to it. But you see over here, what are these thread-like figures? These are actually enzymes. They look like this. You might be very confused what is happening over here. As it says, these are globular proteins. You can see these are globular proteins. Now these curls are basically strains or like chains of protein. You might not recognize these, but they are actually enzymes. Digestion, enzyme controlled process. Now digestion is an enzyme controlled reaction, but it is so important, so I gave it another slide. What food substances we consume are insoluble and non-diffusible, as their molecules are so large they cannot even cross the cell surface membranes. If you remember, a cell has a cell membrane, and if a molecule is so large that it cannot even cross through the channel proteins, if you remember, if you saw my video, then it means it is insoluble and non-diffusible. It cannot necessarily come into the cell. Even it cannot come through endo and exocytosis. It is that large. Rice containing starch along with fish, which contains large chains of proteins and fat, need to be broken down so they can enter cell. For example, a person ate some fish with some rice. Now rice contains some starch and fish contains large chains of proteins and fats. Obviously that is needed to be broken down so that they can enter the cell. They need to be broken down into simpler, smaller, soluble and diffusible substances which can cross the cell membrane. This process is called digestion, whereby large insoluble food molecules are broken down into small, soluble and diffusible molecules, which means they first need to be broken down to enter the cell. If they are not broken down, it will not enter the cell and the body can be malnutritioned, which means it lacks some food, which we will also study in our next chapter, nutrition. Now this is a polymer, which means a macromolecule or a large chain, or for example a carbohydrate, protein or fat. Now it is broken by a process known as hydrolases. We will study that in further chapters, and into monomers, like smaller chains which can be taken into the cell, and they can even be done back. Remember, we did anabolism and catabolism. This is catabolism and this is anabolism because it is made again. Obviously, when a cell needs to create more proteins from the broken down proteins, it will need to make bigger chains of them again. For example, fat which is broken down needs to be used again to in the cell surface membrane, the lipid bilayer, and it will start to do the process of anabolism creation. Enzyme controlled reactions. Now these are three enzyme controlled reactions. Besides digestion, other biological reactions also require enzymes. Given below are some of the processes in a living cell. Obviously, digestion, all those processes, the anabolism and catabolism, breakdown, synthesis, this is done by an enzyme. Synthesis of complex substances from simpler ones like cell proteins by amino acids, which are carried out by the enzymes produced in the cytoplasm. Now, enzymes produced in the cytoplasm basically means produced by the organelles. Synthesis of complex substances. Now this is like anabolism because they are created from simpler to complex substances. This is a ribosome. If you remember in the cell chapter, the first chapter of biology, we studied protein synthesis. Breakdown of complex substances into simpler substances like the oxidation of glucose to release energy from carbon dioxide along with water. This will be done in detail in the respiration chapter. Just know that they can break down complex substances into even simpler substances. For example, the digestion is a huge, and also the basic word of it is catabolism. During some reactions, a poisonous substance, hydrogen peroxide, is produced in the cell. Now, you know there are various chemical reactions occurring in the cell. Now, there is a substance known as hydrogen peroxide which is very poisonous to the cell, that is produced. Now, what will the cell do? Now, the cell creates an enzyme known as catalase that is used to catalyze, which means to break down. Remember this word, use catalyze from now on. Hydrogen peroxide into water and oxygen. The formula of hydrogen peroxide is H2O2, and then oxygen atom is taken away, and then a water and oxygen molecule is formed. Classification of enzymes. Now this is a big thing. Enzymes are classified as to what substances they catalyze, which means what substances they break down. Digestion as an example of hydrolytic reaction. This is called hydrolysis, as hydro means water and lysis means splitting. Now, what is in this hydrolysis is a process where an enzyme breaks down a substance in the presence of water. Hydro means water, lysis means splitting, which means catabolism. That's why these enzymes are called hydrolases. Now, those enzymes which carry out hydrolysis are called hydrolases. Another class of enzymes are oxidation reduction enzymes, which are also called oxidoreductases, which are used in oxidation of glucose during cellular respiration. You can see over here oxidation and reduction. These processes they occur together. And oxidation reduction, also known as redox reactions, we will do in the further coming chapters of chemistry. Other classes include transferases, lyases, isomerases, and ligases. These classes are not so important, but I will cover them right now. Oxidoreductases are involved in the oxidation and reduction or redox reactions. Transferases transfer functional groups. For example, if you know a protein or an amino acid will, for example, have different groups like the hydrophilic group, the amino group, and the phosphate group. These are different chains of molecules attached. They are given the name upon their constituent materials or molecules. Now, for example, an amino group needs to be transferred with another amino group. So the first one I would name A and the second one I would name B. It would be switched. So for this, the enzyme transferases are used. Hydrolases transfer water, that is, they catalyze the hydrolysis of a substrate. Substrate is the substance an enzyme acts on. Lyases add or remove the elements of water, ammonia, or carbon dioxide to or from double bonds, which means if there's a double bond, that will be studied in chemistry. Obviously, a double bond, the lyases can add or remove molecules such as water, ammonia, or carbon dioxide. Now, isomerases catalyze rearrangements of atoms within a molecule, which means, for example, if there is atom A attached to atom B attached to atom C, it will rearrange it to atom C attached to atom A attached to atom B, like mix it up. Obviously that will change the type. Ligases join to molecules. Basically, synthesis is a type of ligase because it joins to molecules. Enzymes end with the suffix ase. You can see reductases, transferases, hydrolases. With this shows what substrate they act on, like carbohydrases, which digest carbohydrates. That's easy. Inactive enzymes have the suffix ogen, like pepsinogen and trypsinogen, which means they are inactive. We will cover what are inactive enzymes further in the limiting factors in part 2, and the prefix pro, like prorenin and prothrombin. This just signifies that they are active or inactive. Extracellular enzymes and intracellular enzymes. Now, what are extracellular enzymes? They leave the cell in which they were produced to exert their functions outside the cell, like the salivary amylase in your saliva that is secreted from the salivary glands from the salivary cells. Intracellular enzymes, they exert their action inside the cells. Obviously, for example, catalase and lysozyme and lysosomes, which break down, for example, the cell also. Examples of hydrolases, carbohydrases, which digest carbohydrates. Now, carbohydrases, we know they digest carbohydrates. There are types of them. Amylases, such as salivary amylase in the mouth and pancreatic amylase in the pancreas, which digest or hydrolyze starch. So, amylases have the job to hydrolyze starch. They are found in the mouth as the salivary amylase and in the pancreas as the pancreatic amylase. The cellulases, which digest cellulose. Though this is not produced in mammals, some bacteria can make this enzyme. As you know, fiber is not digested by our body because it contains cellulose, and we do not have the proper enzyme to hydrolyze it, but some bacteria can produce this enzyme. Proteases, such as trypsin in the small intestine, which digest proteins. That's easy. Proteases, which means those enzymes which digest proteins. And lipases. Now, lipid is another for it is basically another word for fat. So lipases, lipid, such as HPL. Now you might be asking, what is HPL? It is human pancreatic lipase and steps in pancreatic juice. Basically, lipases break down lipases break down fats, and proteases break down proteins. You can see over here, this is an hydrolase. This is a carbohydrate. This is a starch, and it breaking down to glucose. Actually, it breaks down to maltose if it is amylase. Protein. Protease in stomach, it breaks it down into further amino acid, which is a simpler form of proteins, and the lipase in the small intestine breaks it down into glycerol and fatty acids. Now these are the basic elements inside proteins, carbohydrate and fats. Industrial uses of enzymes. This is very important. Enzymes are very useful substances as they are very specific and potent. Now we will learn what is potent in further coming chapter. They are used in industries for several purposes. Yogurt. Microorganisms which are added to milk secrete enzymes which act on it to create yogurt. Now microorganisms are added to milk. We will study this in the chapter of biotechnology in detail, but you should know that microorganisms are added to milk which secrete enzymes which then act to create the yogurt. Cheese. The enzyme rennet is added which curdles milk to create cheese. These are fairly easy examples. Just remember, barley is hydrolyzed by amylases in yeasts which create fermented sugar which are used to create beer. See over here, beer. Barley is hydrolyzed by amylases in yeasts, which means yeast can secrete amylases. Foam rubber. When the enzyme catalase, remember catalase, which breaks down hydrogen peroxide? It will come in handy over here. It is added to latex which contains hydrogen peroxide. It catalyzes the breakdown of hydrogen peroxide to create foam of oxygen bubbles in the latex. So what happens when the catalase is added to latex? It contains hydrogen peroxide. It then breaks it down. Due to this, oxygen which is released in the reaction will create foams inside the latex. This is foam rubber. See, this is foam rubber. It might be familiar to you in the picture. Now, detection of glucose in fluids such as urine. Clinestics, it is a product, contains two enzymes, glucose oxidase and peroxidase. Glucose oxidase catalyzes glucose into hydrogen peroxide. Now glucose oxidase breaks down glucose into hydrogen peroxide, which is then catalyzed by peroxidase, which combines hydrogen peroxide with a dye. Glucose oxidase, now here can be a hydrolase, for example, it breaks down hydrogen peroxide, which is then catalyzed by peroxidase. Now hydrogen peroxide, which was broken down by glucose oxidase, will now be catalyzed by peroxidase. Now what is the job of peroxidase? Peroxidase actually combines the hydrogen peroxide with a dye. Now this tells us it is a ligase. If you see where it says ligase joins two molecules, see, that is done. If there is more glucose, more hydrogen peroxide will obviously be produced, because glucose oxidase will catalyze as much glucose and it will produce all of it to hydrogen peroxide. Now this means that more hydrogen peroxide binds with the dye, which means the peroxidase catalyzes more reactions. Now if more dye binds with the hydrogen peroxide, it means that more intense color of it is seen. The intensity can be measured by a range of colors on the product, which signify the amount of glucose in the fluid. Now this tells us how much amount of glucose is present in the fluid. You can see in this picture, you can match. See, this person's color matches with this color, and it can probably be seen to him, though this is a blurry picture, I cannot see, but it tells us how much glucose is present. This is done in the test for urine, for example, for the athletes which have do running, because if they take some drugs, or like for example steroids, which make them more active or more you can say better in the race, then it means that they are cheating. Now characteristics of enzymes, this is a huge subtopic. Enzymes are very potent. Now what is potent? Potent means as enzyme remains unchanged after the reaction. The meaning of potent, it remains unchanged. They are extremely potent, which means see, when an enzyme catalyzes a reaction, it does not change its chemical composition, which means it can be used over and over again. This shows us the small amounts are enough to bring a large amount of chemical reaction. It has been estimated that one catalase molecule can break down 5 million molecules of hydrogen peroxide in 1 second. Now wait, why not more than that? Because an enzyme does have a, you can say, constant, like there's a limiting factor, because it can catalyze so many reactions, so many reactions, but it is not like infinite. It will run out, but it will first catalyze a lot of reactions. Enzymes and speeds of chemical reactions. Enzymes alter or speed up the rate of chemical reactions that occur in a cell. Now see, without an enzyme, you can see that this is a constant reaction, but it is very slow as compared to when they are enzyme. It speeds our reaction significantly, because obviously it lowers down the activation energy. Enzymes are highly specific. Now what is the meaning of that? Enzymes are highly specific in their action. Amylase will only act on starch and not on proteins or fat, and proteases will only act on protein and not on carbohydrates or fats. The specificity means the specificity means that each chemical reaction which occurs inside a cell will be catalyzed by unique enzymes, which means an enzyme will only act on a specific molecule and no other molecule. The substances on which enzymes act are called substrates, which I told you, like starches and proteins. The specificity of an enzyme depends on its surface configuration, or the 3D structure of its active site. These sites are depression on the surface of an enzyme molecule into which a substrate molecule can fit. Now see over here, before going into the lock and key hypothesis, see over here, this is a shape of an enzyme and this is the substrate. Now this is the active site, this is the depression, and you can see this matches the molecule, which means if this molecule over here comes to this, it cannot be catalyzed or hydrolyzed, depending if it's a hydrolase. It can only catalyze its own substrate, because it will perfectly fit into it. Now when it perfectly fits into it, it breaks it down. But what is lock and key hypothesis? Which means that it acts as a lock and a key, which means the lock can only open one, the key can only open one lock. It is same. See, this molecule can only trigger this enzyme. The enzyme molecule and substrate molecule come closer where the substrate molecule binds or fits into the active site, which is the depression, which complements the substrate, which means it matches the substrate. You see, this fits perfectly into it, and an enzyme substrate complex is formed. So see, when an enzyme is attached to a substrate, this is now known as an enzyme substrate complex. The enzyme then tightens to apply pressure on the substrate, which means you see over here bond breaks under the stress. You see, and over here it sticks together and builds or breaks it by hydrolysis and synthesis respectively to form a new product. Now you should know what is the meaning of hydrolysis. Hydrolysis means that the breaking down of a substance. See, it says over here hydrolysis or breaking down reaction. See, it breaks it down, whereas in this synthesis or building up reaction it creates a product, whereas it breaks the substrate to create a product, whereas it joins the substrates to create a single product. The enzyme binds to another substrate and this process is continued, which means it tells us that it happens again and again and again. Thank you for your support. It has been great to work like this, and thank you, it has been your support that has been making me making more videos for you. Thank you.